Okay, so now an official welcome to everybody. Welcome to everybody joining us from home or from the office. Welcome to everybody in the room. It's very nice to have you on this wonderful, warm spring/summer day. Since we open up as a center to maybe a new kind of audience, and as we've learned from Joacim, the team is growing, maybe a bit of introduction. As I said, please mute your microphones at home. A bit of introduction. So we are the Heidelberg Center for the Environment. My name is Alex Jurka. I'm the executive manager, and we have here with us today one of the managing directors, Thomas Rausch. So we are an interdisciplinary research incubator at the University of Heidelberg, and we connect different disciplines and fields having to do with the environment. And our main focus areas are climate change, biodiversity loss, and the circular bioeconomy. So this is also very interesting for us to sort of get to know your team and the new research group that you're building, and maybe use this opportunity to connect and reach out and see how we could start collaborations in the future. So please feel free afterwards to approach me or Thomas and talk about these possibilities. So that's it for the self-promotion. The main reason we are here for today is for Professor Dr. Joacim Rocklöv, and I've prepared a little bit of bio. I hope I Googled it, so I hope it's okay. You can correct me if it's wrong. So Joacim is an epidemiologist who received the Humboldt professorship at the Heidelberg University, and we're very happy and honored to have him here. And he's a professor for, quote, artificial intelligence in the researching of climate-sensitive infectious diseases. And as I understand, the plan is to explore the influence of climate and the environment on the spread of diseases like malaria, HIV, or dengue. And he is currently working at the Department of Public Health and Clinical Medicine at Umeå University. And here in Heidelberg, he is affiliated with the Institute of Global Health, but we are based in the Mathematikon, as we understood. Yeah. So without further ado, Joacim, again, we're very happy to have you here. Welcome. The floor is yours. You can speak in this microphone so that the people at home can hear you. Yeah. And I will try to manage a bit with the microphones and with the questions if anything comes up. So the floor is yours.
Thank you, Alex, and the Heidelberg Center for Environment for the invitation. So should I just manipulate it? Let's see. Okay, thanks. And it's a little bit awkward almost to have a crowd in reality about this lecture on Zoom. But that's fine. So can you? Yes. This is better. Okay. So thanks for the invitation, and it's really an honor to be here at Heidelberg University, setting up this team and research, which is really important. And I think it's going to be more important in the future with the sort of linkages with zoonotic diseases, infectious diseases, and pandemic preparedness. And I'm going to touch a little bit on that. I thought that's going to be that's a pretty new much work before. More about that later. As Alex said, I'm at the IWR at Mathematikon. I'm originally a mathematician, statistician, but done a lot of work in epidemiology. In addition to the Humboldt project, which just started, I was here first time as a professor a couple of months back. But there's obviously a lot of other projects going on. So I'm heavily involved in a project called Countdown, where we publish a report every year on impacts from climate change on health, different diseases and health outcomes. We also look at the adaptation, how that's developing. We do look at mitigation and co-benefits and economic incentives and communication. So my research and my contributions to life continues to be effective. Talking about the team, there's actually a number of this room here is part of the new labs. If you want to have discussion with them later on, just grab someone in the back. Yeah, so cutting to the chase, I'm going to talk about infectious diseases, and I wanted to start to see if I can change slide. Is this working?
Oh, it's the big one?
It's this one, right?
This one. Okay, so it's upward. I can maybe do it for you. Wait.
Maybe
it's completely blocked now. Oh.
Okay.
It's very slow, I think, but maybe I can, if it doesn't work, I can do it.
I'll try. Okay, so yeah, I just wanted to start from the beginning. There is a lot of infectious diseases. So, and in the sense the title, infectious diseases and climate change and the impacts, it's a huge topic. You know, we can talk about this for weeks. I've been studying a number of infectious diseases and pathogens more in depth, and I'm going to give more in-depth examples, both from a mechanistic perspective, but also from sort of a data perspective on those specific diseases, and to look at, you know, how we generate evidence and what we know about those and the processes and pathways. Emerging infectious diseases is defined as least here, and usually as a disease that hasn't been established in the region as far as we know. There's also re-emerging diseases, which are diseases who've been, you know, maybe eradicated or just not been present for a longer period, but then return. And that's actually the case for, in some places for dengue, it has been established, but then disappeared. Malaria, there's a lot of re-emergence with malaria in terms of because it's been much more widespread in the past than it's currently. There's all the different infectious diseases that we see here, re-emerging or emerging infectious diseases have some kind of specific pathway, of course, how they are transmitted to humans. So we have vector-borne diseases, for example, to be mosquito-borne or insect-borne in general. Tick-borne Lyme disease or tick-borne encephalitis or malaria, mosquito-borne or dengue, for example. There's also airborne diseases, and there's more and more evidence, as you probably know, that the coronaviruses could be aerosol and partly at least airborne. There's also a group of diseases called zoonosis, where there's sort of animal components to the transmission, where we could actually get infected by animals, and animals could get infected by us. We've also seen that with coronaviruses, and as an example, and there's more and more linkage of and more and more data on diseases that are transmitted from animals to humans to animals. And that's definitely going to have much more of in research onwards, just because it's such a it can cause such big problems as we also have seen in the past couple of years. Yeah, and thinking about these diseases that we look at, there's also waterborne diseases, there's different pathways, foodborne diseases. But a lot of them can at least have and show some kind of sensitivity to climate. And what I focus on in my research is trying to figure out, you know, the nature of that sensitivity. And both to some extent from a mechanistic perspective, but also from data-driven perspectives, so we sort of bridge those, you know, the understanding from both domains. We understand both the process, and we understand also, you know, how it plays out on a large scale. Yeah, but that, so the climate sensitivity could be of different types. So if we go to vector-borne disease and mosquito-borne disease, we have the sensitivity of the mosquito to climate conditions. It reproduces, it transmits virus with a different velocity or speed, depending on the climate conditions and the weather conditions. If we go to waterborne disease, it could be more about actually contamination of water from floods or rainfall and that type of pathway, and maybe drinking water or exposure in, if you take swims, there are such diseases like Vibrio as well, where you could have direct exposure, and that would make you sick. Yeah, there's so much, and there's so much climate sensitivity to investigate, and we only know probably a bit of it currently. What we really need to know in terms of infectious diseases is basically how it's related to climate conditions and weather conditions. How climate interacts with other drivers, because if we look at this and understand this realistically, and how it's actually going to, and impacting us and affecting us, both now and in the future, we need to consider drivers and exposure to drivers like climate in the context of other drivers as well. And those drivers could be mobility, it could be vulnerability, susceptibility, it could be a lot of different drivers that sort of co-exist with climate, or sometimes also interact with climate, shaping the exact nature of the outbreak. And that's actually a big knowledge gap, I would say, today in terms of understanding, especially for the future predictions, and to some extent also naturally, because we don't have data for all other drivers in the future in the scenario. So it's a little bit of a gap, but still it's really important to consider because of those interactions and to have realistic scenarios. I'm going to come back to that. A typical question in terms of climate change is trying to figure out what are the future risk areas and how are the risk areas changing, how is it changing in terms of intensity of transmission, how is it changing in terms of number of kilometers or square kilometers of land area in the region that becomes susceptible to outbreak, yearly or so, or for specific time periods, 2020 to 2050. Or that's very important for adaptation and for preparedness to cope with those outbreaks, of course. Another question, which is also interesting, is can we use, because there's a wealth of climate information, means we can do projections, it means that we can potentially use climate information like subseasonal forecasts of climate to predict diseases. And if we can do that, we can predict disease burdens or outbreaks or incidents ahead of time. We also have lead times in our disease. So that would be really quite important if that works out well. And it looks like it could actually be working out well for certain diseases, that we have enough good predictive ability to actually operationalize that. So there's quite a lot of work on danger, for example, in terms of subseasonal forecasting early warnings. There's an operational early warning system for vibriosis. Vibrio is a waterborne disease that's common in the Baltic, run by the European Centre for Disease Control and Prevention, as an example. There is so much data coming available from the climatology and meteorology side, and also from observations, etc., that could just be very useful in terms of public health. It hasn't in the past been so very useful because there hasn't been the research to support it, and there hasn't been the local capacity, the institutional capacity to actually do something about it. Okay, so where do we know more about disease? We know more about diseases in the group of vector-borne diseases. There's more research in that domain. Quite a lot specifically on malaria and dengue. There's also quite a bit of research on tick-borne diseases. We also know a little bit more about, the state of the art, and the evidence is better in terms of waterborne diseases. And there's quite a lot of studies that show relationships between cholera and Vibrio and drinking water associated diseases as examples. That's not exclusive, there's more. But then we know much less about diseases which are mediated through some kind of animal reservoir and maybe even animal insect human transmission. I'll give a few examples, and also I'm going to show an interesting study that came out last week. Okay, so the pathways to human outbreaks that's really, I think, important are described in this graphic. We have environment and climate as as a sort of a causative factor, and other drivers. Other drivers to be mobility and trade. This could be a lot of different things. It depends on the specific disease and the pathway. We also obviously have these direct or more or less direct pathways where we have water or foodborne diseases causing outbreaks and transmission. And we also have the pathways through insects and wild and domesticated animals. So those obviously, wild animals do carry a lot of viruses. Bats have been up on the agenda a number of times the last couple of years, but also monkeys and other animals, chickens, obviously with the avian flu, and wild birds and domesticated birds. And they also transmit disease from each other when the viruses are more or less similar, so you can sort of spill over from one species to another. Mosquitoes and insects that also help with those sort of spillovers or sharing of viruses from one species to another. And they could also, they could obviously also transmit directly from humans to humans by a mosquito like malaria and so on. But there's also situations where you have animals and ticks, for example, Lyme disease, and the tick get infected by the reservoir animals and then bites a human, and the human gets affected. And that's the system. So the system that you would need to look at if you want to understand in detail, such a disease would be a system where you can actually describe the wild or domesticated animal and transmission among the animal, and how it interacts with maybe a breed vector or just directly to humans. And that's a big, I think, gap in terms of what we know of today. And why is that important? Because environment is going to, and climate is going to cause changes among those animals and insects as well, of course. And if we only look at the direct pathways between environment and climate, we might actually miss out important changes in mediating that impact in how wild animals find new areas to invade, or maybe how wild animals are actually decreasing or increasing their densities and so on. Yeah, okay. So and through all these different sort of parts of the pathway, there's preparedness, policy, and interventions, of course, and social conditions that sort of can change and intervene. So there's a lot of environmental and climate policy now, greening, the Green Deal in the EU and so on. That's obviously going to change a little bit the environment and the situation in cities, blue cities, green cities, and so on. There's also a lot of conservation going on, which is going to have impacts, both probably intended impacts and unintended impacts. It's really important to look at that. There is also a lot of health policy and health policy and preparedness that could, for example, contribute to building surveillance systems and rapid and early detection of circulation of pathogens. And that could be both. That could be both in humans and human surveillance systems. We have, I mean, it's not good, it's quite decent anyway. But when we go to surveillance systems in, you know, for mosquitoes or for animals, and especially also animals and mosquitoes with, you know, infected with virus, then the data is much more sort of sporadic and sketchy. But there is some data and there is much more generated. Yeah. Okay, so just a few examples I wanted to mention is this is a good overview on some of the infectious diseases and the other diseases from climate change. And there is always a report published in the fall. From this year, there will also be, so in the fall this year, there will also be a new report launched counted for Europe with much more detailed data and many more indicators and disease indicators in for example, in the infectious disease domain. So that would be also a sort of a continuous activity that the group, my group, contribute to, developing indicators, validating indicators, and updating indicators through their publications. A lot of the focus with the last countdown is actually on policy and advocacy, but there's a lot of rigorous research behind each one of those indicators in the report, and so much effort going into this report. So this report will be something to keep an eye on when it will be launched, I think. You know. Okay, so let's talk about some specific situations and sort of see if we can bridge these sort of mechanistic statistical relationships in understanding for one disease. I'll start with dengue. I've been working a lot on dengue, and it's a big disease. The estimates a couple of years back, or I think probably 9 years back, the global burden of disease estimate was sort of 400 million infections a year, but a lot of them not really symptomatic. So about 100 probably, you know, symptomatic infections. So it's a big disease. It's not killing a lot of people nowadays, at least. But people can get severely ill and need clinical care, and you could also have a little bit of long-term problems after infection. Worst case, you have a hemorrhage, so sort of, you know, bleeding inside, and that could be really bad. So the problem with dengue is it's not so much the number of deaths, it's actually rather that the disease epidemics are occurring at the same, everyone gets sick basically at the same time. You have this extreme burden on the healthcare. So it's in that sense, it's still a really political disease, a little bit. We understand it better now, I think, for us here, because of COVID as well. It's a big strain on the healthcare services during epidemics, and obviously the care could also be severely constrained if it's really big outbreak. So dengue has definitely a strong climate component in its sort of as one of the drivers, and looking from a mechanistic perspective, and this is one of the indicators in the Lancet Countdown report. We could look at the R0 value. So the R0 value is basically the basic reproduction number. It tells us how many new cases you expect out of one infected or infectious person in a totally naive population. And you can see here how climate and the warming basically has sort of driven a change in the basic reproduction number over time. Now, the basic reproduction number is not totally valid in all, this is a global aggregate measure, it's not totally valid in all situations because you have some immunity, so that's not incorporated. This is just purely how the potential of the spread is catalyzed by the warming. And I'd compare it to transmissibility change. So in a sense, climate is contributing to transmissibility change in a disease like dengue, because you have a more rapid replication of the virus of the mosquito and a more effective transmission. Yeah, and it's so far, between 5 and 10% according to the model prediction. And every year there's kind of an either or. It's a pretty steady trend. There's two different mosquitoes described here. There's Aedes aegypti and there are Aedes albopictus. Aedes albopictus, I think, is here, right? Probably. It's been seen here, I think, at least. I don't know if it's totally established. It is. So we could probably go out for expedition next time.
The warming is Stadtblatt and Heidelberg about this Heidelberg.
Okay. Yeah, so it's been rapidly expanding in Europe since the 90s when it was introduced to trade. It's more sort of capable of coping with colder winters than Aedes aegypti. Aedes aegypti is a more tropical mosquito, both transmit dengue, chikungunya, Zika, yellow fever, etc. And Aedes albopictus is a little bit less competent, but it is competent, and it actually drives outbreaks even in countries like Sri Lanka has a lot of Aedes albopictus, and that's a big source of their over the last 20 years, it's been expanding, so one could actually see every year almost from the surveillance, mosquito surveillance data, how the mosquito distribution has changed. And that's obviously partly driven by climate, but it's also partly driven by mobility and trade, because mosquitoes doesn't fly that far. They would probably not fly more than a couple of 100 meters, usually, normally. But they could also take a ride with a truck or the car and go somewhere later in a new place. So there's like a fast invasion from motor vehicles, and there's a slow invasion, which is more like gradual every year, which is more natural mosquito expansion. Okay, so why can it survive the winters? Because it can die for, so it can lay their eggs, and the eggs can survive even if it's snow and cold. And when it rains in the spring, the eggs will hatch, and there will be mosquitoes. Yeah, so that's really important, and that's not really the case for Aedes aegypti. There is no diapause in sort of like that. Otherwise, mosquitoes have sort of a usually life cycle where there's egg laying, there's larva, there's pupa, and then there's another mosquitoes. And all those mosquitoes, it's only the females that do search for blood meals because they need that for their egg laying. So that's basically it's a quite complex cycle. It has a lot of sensitivity to climate and dependence on climate and temperature, both in development in the different stages. You see survival for eggs, it depends on temperature and rain, and larvae and pupa stages, etc. And that can obviously be incorporated into models. And here is just like a very simple mosquito model described with different mortality rates for different stages of the life cycle and so on. and dependencies to avian conditions. Additionally, to looking at the mosquito life cycle, which sort of governs the population dynamics of the mosquito, you could look at vectorial capacity. Vectorial capacity is sort of an old thing that's been hanging in the field for a long time. Mosquito modeling was introduced by from malaria researchers more than 100 years back. Prof and McDonald, etc. And more or less the same rules would guide the models today. And the basic reproduction number is related to the vectorial capacity. This is a little bit technical. The vectorial capacity is depending on the mosquito density, the biting rate, which is A, the vector competence, so basically how competent is the mosquito to actually get infected and be able to infect others. So replicating virus and becoming infectious itself. So you sort of have a virus in saliva. And then the extrinsic incubation period. So we have an intrinsic incubation period when we get sick. It takes time for us to develop the virus or a disease to become infectious. The mosquitoes are the same. They have an intrinsic incubation, extrinsic incubation. And what's really important when we look at insect-borne diseases, and especially mosquito-borne diseases, is that if it's cold, the mosquitoes will survive a very long time. So they won't have enough time to actually develop infectiousness. So it's not only really that they might exist in a location and they might reproduce, but they might not have the survival length to actually spread disease. So vectorial capacity is also very important in addition to just mosquito population dynamics. Here's just two graphical illustrations of how the vectorial capacity depends on the mean temperature, the daily mean temperature, and the daily dynamic range of temperatures. And it basically shows that they have the highest probability or likely vectorial capacity to transmit and diseases when it's stable conditions, so it's low variability over the day, and it's around 30 degrees. So that would be typically, Sri Lanka or Thailand or some of those countries where it's quite stable and usually also humid. And when you go to a place like Italy or Heidelberg, you have a much higher dynamic range, so you usually get much lower darkness.
Should I try from here?
Yeah, let's see if it works. Take a sip of water.
I don't know why it happens.
Too many images, maybe. So the next question is. Yeah. Good. Thank you. Okay, so the next question is, how well does this model work? Well, the models work reasonably well. Actually, here's just a model validation of a model to data from Reunion, where we had surveillance data on mosquito activity and then made predictions. It's not perfect, but it does capture the temporal dynamics to some extent, and also, if we look at geographical patterns, it does capture geographical patterns quite well as well. So it's possible. It's probably possible to do better models, but it's still reasonably good in terms of validity. Based on that, we could also obviously also ask questions. We looked at the trends up to now in terms of vectorial capacity or not before. We could also think about what's going to happen in the future, and because there is a lot of climate data available in Copernicus platforms or through different climate projects, we could plug that into the models to run projections and see as experiments what if the climate forms according to a certain scenario. Here we're using a number of different scenarios. We're using different carbon concentration scenarios, RCPs, representative concentration pathways. The ones here, the blue one is the 2.8 and the red one is 8.5. So basically, the worst red and the best scenario. And it looks quite different in the future, especially there's quite much more potential for mosquito populations and dynamics in the high emission scenario. This is a global aggregated figure. It's actually run at a much wider scale, 50 by 50 kilometer in this example. And that's seen here. So here you can see the distribution in the intensity of mosquito, predicted intensity of mosquitoes in the end of 21st century in response to a low emission and concentration future and a high CO2 concentration future. And especially, I think this is Aedes aegypti, so it's more tropical mosquito. Especially US, there is big changes. In Africa, there's big changes. And in Europe, there's also quite some changes, actually China. There's obviously always going to be situations where you also have contractions, just it's too hot. Mosquitoes wouldn't really thrive if it's too hot. Dengue mosquitoes seem to be more resistant though, to very hot conditions compared to malaria. So if you compare malaria and dengue impact projections for the future, you usually see that malaria drops down faster than dengue. It's more sensitive to very hot conditions, especially malaria is more distributed in sub-Saharan Africa as well, and some areas that will definitely get too hot. Although that's going to, if you look at populations increase, you see increases still, but not huge increases, but it's going to be different populations. And there's a complex dynamic in terms of semi-immunity. So if you haven't had malaria circulating a lot in the past, you're probably going to have more severe outbreaks as well. So it's also important to consider. I wanted to mention that we are also working in a project called EasyMit, where we are trying to work with the community of epidemiologists and help people to contribute to producing comparable impact estimates. If you do mosquito impact estimates on malaria or dengue, if everyone would do it in a similar fashion, it's going to be possible for us to compare across models, for example, impact models and studies. And if everyone else in the other sectors, maybe the other health domains, or in the cultural, or forestry, or biomes, or water sectors, also using the same protocol, then we can go from comparing one disease to a whole range of sectors in a comparable and consistent fashion. So this is a project that's led by the Prof Institute for Climate Impact Research, and we're contributing. I'm contributing as sort of coordinator. There's quite a big number of activities in this project. From a totally different perspective, if we go now, I was talking about the mechanistic pathway before. If we look at just at the data instead, and see what can we do from based on data. Here is dengue outbreaks in different parts of the city of BRÜCKE in Indonesia. So here is the city. It's 44 villages in the city, and here's the time series basically of events and outbreak events in the city over time. Then to study this data, we had climate data, we had weather data, weather conditions for the city. We also collected mobility data and mobility data from social media. Usually what one would do in statistical or spatial temporal study would be to have some kind of covariance structure that tells you if one village would be more related to another or less related to another. And that you could also have over time to some extent, in time and space. But that's not very useful. We know something about the transmission here. We know that climate is actually making mosquitoes capable of replicating virus and also to grow in numbers. But we also know that humans are carrying the virus from one location to another. So mosquitoes need humans in order to transmit, right? So for a new outbreak to take place in one village, in another village of the city, you need both mosquitoes present and you need people coming there with virus. So, and how do they come to these places with virus? Well, they've been in an area with an outbreak, and there's a latency there. So basically what we did is a little bit unclear here in this figure. We took this mobility and constructed an adjacency matrix, so basically telling you how related the different villages were to each other. And based on that and the climate information, we could do much better predictions. We used the adjacency matrix as a covariance structure. And that worked really much, it worked much better than there is a lot of studies using climate weather conditions before. But this worked much better. We could predict to some extent at least, and with quite high accuracy, capture the actual incidence in the conflicts intervals of the models for all to some extent. And so basically that shows that, you know, just looking at the data, maybe you could do better predictions on incidence and transmission dynamics. And maybe that could also be used for us to understand better the potential future dynamics. It could definitely help disease management if we could produce warning systems incorporating both weather dynamics and dynamics. I want to mention something else as well. I have a little bit time left, or is it half an hour? Okay. So, okay, very little. I wanted to mention West Nile. That's also a very relevant disease for this region. It's a mosquito bird disease. Basically, birds bring virus through their migration routes, and mosquitoes bite birds and they bite humans and cause outbreaks. In short, so it's basically the migratory patterns. There's been big outbreaks in Europe the last years. We did a study here on West Nile, and the interesting thing was, can we predict the big outbreak of West Nile 2018? It was much larger than before. We started collecting data, and we had data for previous years. We had data for 2010 to 2017 for different parts of Europe in collaboration with ECDC. We started calibrating models and actually managed quite well to predict the 2018 outbreak and geographical expansion in the 2018 outbreak. And the most important variables into the model for the predictive model is actually climate and bioclimatics. So bioclimatics also have 1 quarter and an indicator of weather conditions in that quarter. We had the satellite sort of normalized water index as well, and vectors and birds in the north. But climate is really important, and in the previous slide as well, was shown that the West Nile is actually really expanding further. There are sites of West Nile cases in new intentions also after this. is likely to be affected by climate warming. A last mention, and this is only one for start. This is really interesting study that came out 1 or 2 weeks back. It's just sort of advanced press in Nature at the moment. It looks at viral sharing across mammals and how that's affected by climate change. So it basically runs a lot of models to estimate how climate change is affecting mammals and how, based on data on what viruses and pathogens mammals are usually carrying and the similarity between mammals in their sort of susceptibility, it estimates if it's likely that you have viral spillover sharing between those animals when they change their sort of niche or look for new areas to sort of evade or just live in. Basically the finding is that the main finding is that the cross-species transmission is likely to increase a lot according to these models and simulations up to 2070. It's not so much depending on if it's a high warming trajectory, if it's a low warming, it's quite strong anyway. But it's a big change. There is likely to be, according to this study, quite a lot of much more transmission between different mammals that we wouldn't expect without warming. And that's also to some extent overlaying with locations where there's big population numbers. So that could potentially, maybe likely lead to new outbreaks of zoonosis and pathogens. That's the last thing.
Thank you very much, Joacim. I'm just going to stop sharing. I don't. And for those of you at home who are going to watch this in the future, thank you for listening. I will stop the recording right now.